So, Neta, for coming from so far, from the Faculty of Medicine here at Tel Aviv University. Neta is, I need this.
I can't without mine.
Neta is the vice president of the Israeli Society for Cancer Research. She is a board member of Metastasis Research Society, the MRS. She's chair of the Department of Pathology, and the past chair, just recently
changed.
Came the past chair. And her lab is studying the link between inflammation and cancer, and we're really happy to have you here with us today. Thank you.
Okay. So maybe it's good to have them around. Thank you very much for inviting me for this wonderful day. It's so good to be in an in vivo conference after so long. And I'll tell you today how stromal and immune plasticity shape the metastatic microenvironment. So it's very, we haven't heard a lot today about stromal cells, but we did get the impression that tumors are multicellular organs, and we're no longer treating them as just a bunch of cancer cells. We know that cells of the immune system, stromal cells, blood vessels, all take a very important part in all stages of tumor progression and metastasis. And this meeting started with looking at statistics of cancer mortality. It's important to say that cancer mortality is almost exclusively a result of tumor metastasis. So the data that you see here is from breast cancer, but this is also true for many other cancer types, where the survival goes dramatically down if tumors are diagnosed after cells have already disseminated. So like I said, this is from breast cancer. And the reason for that is because advanced metastatic cancers are largely incurable today. We can prolong life to a certain extent, but they're usually not curable. And we have limited knowledge on the early stages of metastasis formation, which is why we believe that understanding the biology of tumor metastasis is the most urgent question of cancer research today, and this is what we're focusing on. So going back to the tumor microenvironment, there's several decades of work, and we accumulated a lot of knowledge on the role of the microenvironment at the primary tumor site, but the role of the metastatic microenvironment is still largely unresolved. I mean, it's a topic of a lot of research. And we do know that changes in the metastatic niche precede metastasis formation, but in human patients, these early events of metastasis are usually a black box, because this is when we still do not know that metastasis is happening. So basically all the drama is happening when we don't know that the metastases are there. And studying these early stages, or studying metastasis in mice, was also hindered by the fact that there were not a lot of good models to show the multi-stage process of metastasis. So these are the things that we've been trying to tackle and really uncover the biology of tumor metastasis, and we've been doing this in two major arms, looking at cancer-associated fibroblasts and immune cells in breast cancer, and at neuroinflammation in melanoma brain metastasis. These are now converging because we're also looking at breast cancer metastasis to brain. But the main questions in what I'm going to tell you today is we were asking how is an inflammatory microenvironment instigated in the metastatic niche, and what is the functional role of stromal and immune cells in facilitating metastasis. So this whole thing started a few years ago with a project initiated by Dr. Yael Raz, who's also a gynecologist. She's now at Ichilov. And we used genetically engineered mice that express YFP under the collagen 1 promoter, which enable us to isolate in an unbiased way their fibroblasts, and we crossed them with PYMT, transgenic mice that develop breast cancer. So now we had all cancer-associated fibroblasts or normal fibroblasts labeled with YFP. And we isolated fibroblasts from lung, either normal lungs or lung metastasis at early and late stages, and we profiled their transcriptome. I'm not going to tell you the whole story, but I'm really cutting a very long story short. But what we found is that fibroblasts expressed distinct and stage-specific gene signatures, so they clustered separately from micrometastasis and macrometastasis, suggesting that different transcriptional programs are operative at different stages. And we analyzed all these different pathways and also the transcription factors that were the drivers of this transcriptional plasticity. And we found that at the stage of micrometastasis, ECM remodeling and stress responses were the most highly upregulated. And later on, we had ECM remodeling and inflammation. And I'm going to focus a little bit on what we found in the proinflammatory signaling that is upregulated in fibroblasts. So this is a story that was led by Ofir Shani, an MD PhD student in the lab. She now graduated. And when we analyzed the RNA-seq data, we found that IL-33 was upregulated in metastasis-associated fibroblasts. It was one of the most highly upregulated. And when we validated this, looking at supernatant that we made from normal lungs or from lungs with metastasis, and here we did everything in parallel in a transgenic mouse model of spontaneous metastasis, and also in a transplantable model. So in this model, the mice are injected orthotopically with 41 cells, triple-negative breast cancer cells. We resect the primary tumor and then wait for spontaneous metastasis to occur. And we also looked at bronchoalveolar lavage fluid, giving us a snapshot of secreted factors in the lungs. And in both cases, I hope you can see that IL-33 was highly upregulated. So what is IL-33? It's a member of the interleukin-1 family. It plays an important role in tissue homeostasis, in inflammation. It's a proinflammatory. It's an alarmin. So cells secrete it when in response to stress. And the cells that respond to it are usually immune cells, mostly type 2 immune cells. So it's a driver of type 2 immunity. So we made sure that indeed fibroblasts were actually the main source of IL-33 in metastatic lungs. We isolated different cell types, immune cells, endothelial cells, and fibroblasts. And what we epithelial cells also, and what we saw was indeed that fibroblasts were the main source of IL-33. We also saw this by staining, so IL-33 is in red, and fibroblasts are in green. So you see that most of the cells that express IL-33, so not all fibroblasts expressed IL-33, but IL-33 was mainly expressed by fibroblasts. And we also, when we analyzed the metastatic burden of these mice, we saw that IL-33 expression was in correlation with a higher metastatic burden. So we went ahead to do a functional assay. Again, I'm cutting a long story short. So these mice were treated with anti-IL-33 after resection of the primary tumor. And then when we analyzed and quantified their metastasis, both by CT and also by H&E staining, we saw that mice that were treated with anti-IL-33 had attenuated lung metastasis. And when we looked at the mechanism, we looked at different, like I said, IL-33 drives type 2 immunity, so we looked at different cytokines and chemokines that are associated with type 2 immunity, and we saw that the mice that were treated with anti-IL-33 had attenuated expression of all these type 2 cytokines and chemokines. And when we looked at the recruitment or the presence, we did immunophenotyping of these mice, we saw that there was also an inhibition of the recruitment of different immune cells to the lung metastatic niche. And we try to validate our findings in human data. We work with mouse models, but we try to remember that these are only models and to see that our results are actually also relevant to human disease. And when we looked at data sets of breast cancer metastasis to different organs, we saw that IL-33 was actually upregulated in the stromal compartment of lung metastasis of breast cancer, suggesting that maybe it is something that is organ-specific. And then in collaboration with Iris Barshak from Sheba, we managed to get a cohort of tissue samples from human patients with lung metastasis from breast cancer, and we stained for IL-33, and I hope you can appreciate here the stromal staining of IL-33, really validating our results from mice, and the fact that IL-33 was upregulated in lung metastasis, but not in normal lungs. So what we showed in this story is that CAF-derived, CAF cancer-associated fibroblast-derived IL-33 drives a metastasis-permissive microenvironment in lungs by activation of type 2 immunity. And then switching to a different story, I want to talk a little bit about chemoresistance. So resistance to therapy is also a main factor of metastatic relapse. And when you look online, you do Google images, you look online for different illustrations of chemoresistance. Most of the things you find, for example, like this one, talk about intrinsic factors of the tumor cells. How they become clonal selection and changes in the tumor cells. But in recent years, there's quite a few studies that show that the microenvironment also plays a role in facilitating and mediating chemoresistance. And we know that actually there are paradoxical effects of chemotherapy. So in Danny Pear's cartoon, in the movie that he showed us, chemotherapy looked like carpet bombing. So there's always a lot of collateral damage. And indeed, it's a double-edged sword. It can efficiently kill tumor cells, but there's also, it causes tissue damage and induces pro-inflammatory responses that actually support the survival and dissemination of cancer cells. So in this project that I'll show you in the remaining few minutes, we found that CAFs mediate chemoresistance and lung metastatic relapse, and this project was led by two outstanding students, Lea Monteran, a PhD student in the lab, and Noor El-Sheikh, who is already an MD, PhD, and a resident, but he's still doing research because he's such a passionate scientist. And actually, when Noor was a PhD student in the lab, he showed that fibroblasts can sense tissue damage associated molecular patterns, by activation of the NLRP3 inflammasome. And since chemotherapy also induces a lot of tissue damage, we thought that maybe fibroblasts will also be sensing this and inducing pro-inflammatory signaling in response to chemotherapy. And this can either be a direct effect or in response to dying cells. So this was the thing that Noor and Lea set out to find. And they started actually in a system without cancer. They just treated mice systemically with chemotherapy, in this case doxorubicin and cisplatin. And then they isolated fibroblasts and looked at the expression of pro-inflammatory cytokines and chemokines, and they saw that systemic chemotherapy really induced inflammation in a cancer-free system, and it also induced recruitment of different immune cells into the lungs of mice that were treated systemically. And they also did an experimental metastasis of mice that were first treated with chemotherapy. So these mice did not have cancer, treated with chemotherapy, and then injected with cancer cells IV. And what they found is that chemotherapy actually preconditioned these mice, and the mice that were treated had enhanced lung colonization. So at this point, we decided to look at these things in a more physiological system, and we switched to a setting of adjuvant treatment, which of course mimics the clinical situation better. So these mice are injected with cancer cells. The primary tumor is removed, and then they are treated with adjuvant chemotherapy, and we're following up for metastasis. And what we saw is that adjuvant treatment, actually, in this case, doxorubicin was inefficient in reducing lung metastasis, but cisplatin significantly attenuated lung metastasis. And when we looked deeper into what happens at the earlier stages in mice that were treated with doxorubicin, we saw that the treatment actually disrupted the immune milieu. You can see here a lot more granulocytes in doxorubicin-treated mice, but cisplatin actually was associated with more T cells. And mice that were treated with doxorubicin had already at early stages more micrometastasis in their lungs. When we quantified it, this is just showing the difference in immune cells. So doxorubicin enhanced the infiltration of granulocytes and monocytes, whereas cisplatin enhanced infiltration of T cells. And when we looked at the granulocytes that were actually recruited to mice, we saw that they were expressing a signature of an immunosuppressive signature. Both the myeloid cells and the granulocyte cells. So doxorubicin treatment actually increased the recruitment of granulocytes and monocytes, but also enhanced their immunosuppressive gene signature. And we tested this in vitro to see whether they actually have suppressive activity. So this is a T-cell proliferation assay. We co-cultured these cells that we isolated from treated mice with T-cells. And what we found is that MDSCs that were isolated from doxorubicin-treated mice were more efficient in inhibiting T-cell proliferation than the controls. And we also saw that mice that were treated with doxorubicin, and we looked both at early and late stages of metastasis, enhanced the accumulation of T cells that were expressing immune checkpoint receptors. We looked at TIM3, PD-1, and TIGIT, and this was true both at early and late stages. So we have pro-inflammatory and also immunosuppressive microenvironment. So just to summarize this, we see that treatment with doxorubicin enhanced granulocyte and monocyte recruitment, enhanced their immunosuppressive activity, the dysfunction markers on T cells, and it failed to inhibit lung metastasis. But I promised you a story about fibroblasts. So we looked how does chemotherapy affect lung fibroblasts and how they affect lung metastasis. When we isolated fibroblasts from treated mice, and we profiled their gene expression, we did RNA-seq and nanostring analysis. The first thing we noticed is that their gene signatures were actually significantly different than normal fibroblasts. And when we further analyzed, again, cutting a long story short, we were surprised to find that the main pathway that was upregulated in fibroblasts in mice that were treated with adjuvant chemotherapy was the complement pathway. Again, we made sure that the fibroblasts are indeed the source by isolating different cells, and we saw that fibroblasts are the major source of complement component in the lung microenvironment. Then we looked at a lot of several available human data sets of expression of single-cell RNA and RNA-seq from lung primary tumors and from lung metastasis. And what we found also in this case was that lung fibroblasts were a major source of complement components in lung malignancies. And we looked at the different components, and we again saw that this is increased in lung metastasis, same thing that we saw with IL-33. So organ-specific metastatic microenvironment have different pathways that support metastasis. Just a really quick reminder of what the complement system is. So it's a component of the innate immune response in response to pathogens and tissue damage, but it's a bridge to adaptive immunity. Activation of complement system is highly conserved for sensing dangerous signals, and it was also shown to be involved in different aspects of cancer progression. And an important thing is that C3a and C5a, two components of the complement system, are powerful chemoattractants. So we thought maybe calf-derived complement and chemoresistance is related to this. We looked at we compiled a general complement score, so looking at all the components of the complement system together. And what we found is that the expression of complement system correlated both with metastasis and with immune suppression in lung metastasis, suggesting that it may play a role. And indeed, we saw that C3a is increased in the lung microenvironment following doxorubicin treatment. When we saw that this is the case in early and also in late metastasis when we isolated lung supernatant. So we just looked at all the secreted factors in lungs, and we saw high levels of these. So we hypothesized that maybe calf-derived complement signaling facilitates immune cell recruitment and their functional reprogramming. So we went ahead and did a functional experiment. These are my final slides. So we used two blockers, one of the C3a receptor and one of the C5a receptor in combination with doxorubicin treatment. So again, this is in an adjuvant setting following removal of the primary tumor. And then when we looked at early metastatic stage, we saw that complement blockade in combination with doxorubicin reduced the immune cell infiltration and the expression of T-cell checkpoint molecules. And of course, the most important thing, we looked at the metastatic burden, and we saw that combining complement signaling inhibitors together with doxorubicin reversed the adverse effect of doxorubicin and made it more efficient in this system. So just to summarize what I showed you here, this paper is under review. So if our reviewers are listening, we've been waiting since December. Please send us back your reviews. What our results show is that treatment with chemotherapy probably induces damage-associated molecular patterns and cell death that activate pro-inflammatory signaling in fibroblasts, and they start expressing complement system recruiting immune cells that express the receptor. And by combining chemotherapy with complement signaling blockade, it may be beneficial in preventing the adverse effects of chemotherapy. So this is our team. Nu is not in this picture, but Nu and Leah really led this project beautifully. And these are our collaborators and our funding sources, including the CBRC. So that's it, and thank you very much for listening.
Thank you, Neta, for the great talk, as always. I think that the work has several worrisome implications, of course, for the clinical world. Thinking a bit reminded me of the work of Yuval Shaked when what he saw with paclitaxel and induction of angiogenesis and metastasis. So I was wondering, how relevant is it to patients? What do we know from patients being treated routinely with doxorubicin, as you mentioned, and some other chemotherapeutic drugs, and the induction of metastasis? And do you see it being one of the reasons of non-responders to immunotherapy as well, if we think about that, combinations that are routinely given?
So it's obviously an excellent question, we tried to answer this question throughout this project. It was a very tricky one to answer because, as you know, and as you said, doxorubicin is used routinely for treatment of breast cancer patients, and some of them respond and some of them relapse. Cisplatin is actually used sometimes as a second line therapy, and in our hands it was more efficient. But we really wanted to answer this question whether there is complement signaling or whether we can get some kind of snapshot, but the available samples, or even just to do the statistics. So we did this. You see, one of our collaborators is Amir Sonnenblick from Ichilov. He's the head of the breast cancer unit at Ichilov, and it was really hard to obtain any relevant data that could help us analyze this systematically to answer whether complement signaling or fibroblast activation are upregulated in patients following doxorubicin treatment. I can elaborate later on all our heroic efforts to get this data. So, great question. I don't have data to answer it, unfortunately.
So really related to what Ronit asked, do you have a biomarker for this specific type of complement activation? As complement does circulate, maybe it will be easier than usual to find such a biomarker. And then you would be able to identify these patients, because tissue samples you're not going to get. They're not biopsies at this stage.
Okay, so I will get into the details because I see that this is the question. So what we see, what we see in mice and also in human, is that the blood levels of complement are not predictive. So what we did in mice is look at the bronchoalveolar lavage fluid to get a snapshot of what is happening in the lungs, because the systemic levels, so it was not a problem to get blood samples of patients. Not always, usually not right after they got chemotherapy, so not at the right time, but we could, in principle, design a prospective study and collect blood, but it's not there. So the only way to do it is to look at their at their BALF, at their bronchoalveolar lavage fluid, and that I will tell you later. We tried that as well. It's really difficult because we. Well, we tried.
Any other question? Yes, just a second, take the microphone.
So thank you for a great talk. Just a quick observational question. So, I mean, we're discussing more and more immunotherapies, but you chose, and that's very courageous, to go and look also at chemotherapies that we all know are still being widely used. Do you think there's a stage at which will we always have the two combined, meaning immunotherapy and chemotherapies? And more than that, do you think there's a certain stage where an immunotherapy alone may not already be capable? The body itself can't work against a tumor, and you actually need help, for example, from a chemotherapy. How do you see it as a researcher in the field?
Tough question. So overall, I think combinations are the best, and I think in the immunotherapy world, and we also see that usually just treating one target is not enough. And I think this is also what we showed, that if you combine doxorubicin with something that modulates the immune system, then you get a better outcome. So along the same lines, maybe a combination of chemotherapy with checkpoint inhibition, but these are things that are being, being tested and have been tested, and there is a lot of data on that. So we're not going in this direction simply because what we found, we started looking at fibroblasts, and then we found ourselves working with complement system. So